In a 2023 FDA approval, a novel topical foam hit the dermatology market. Marketed as Zoryve, it was formulated specifically for the treatment of seborrheic dermatitis. The active ingredient driving this clearance is Roflumilast, a highly specific phosphodiesterase 4, or PDE4, inhibitor. But the foundational proof of concept for this molecule didn't happen on the surface of the skin. It happened in a cohort with failing lungs, specifically in one of the most difficult-to-treat patient populations in medicine, severe chronic obstructive pulmonary disease. This creates a fascinating pharmacological case study. We have a single mechanism of action translating across two drastically different disease states, entirely dictated by altering its route of delivery. To understand how this drug safely clears the skin today, we must first analyze how it fought treatment-resistant inflammation in failing lungs back in 2015. In severe COPD, the standard treatment paradigm relies on a combination of inhaled corticosteroids and long-acting bronchodilators. But this approach has a physiological limit. This line chart illustrates a flat dose response curve. Pushing higher doses of inhaled therapies eventually stops yielding better inflammation control. The benefit simply plateaus. Worse, increasing those inhaled corticosteroid doses actively raises the patient's risk of developing pneumonia. This creates an exacerbation ceiling. Severe patients hit a wall where they are maximally medicated, yet continue to suffer periodic, dangerous respiratory events. Breaking through that ceiling safely requires an orthogonal, systemic mechanism of action, something that operates outside the boundaries of standard inhalers. The REACT trial was designed to test exactly that. It was a rigorous, one-year, double-blind, placebo-controlled study, drawing patients from 203 global centers. The inclusion criteria were stringent. Patients needed a documented history of severe exacerbations and ongoing symptoms of chronic bronchitis. Looking at Table 1, we see how heavily medicated this cohort was at baseline. An overwhelming 98% were actively taking an inhaled corticosteroid and long-acting beta-agonist combination. Furthermore, 70% were managed on triple therapy, adding long-acting muscarinic antagonists. Despite this massive pharmacological blockade, these patients were still landing in the hospital with frequent exacerbations. This population represented the absolute endpoint of inhaled therapy. Any statistical improvement seen here would prove the power of systemic PDE4 inhibition. The primary outcome researchers sought was a reduction in the rate of moderate to severe COPD exacerbations per patient per year. During the trial, the placebo group experienced fewer events than statistically anticipated. This required researchers to run a predefined negative binomial regression sensitivity analysis to ensure the findings were robust against distributional assumptions. Figure 2 plots these results, revealing an initial 13.2 to 14.2% drop in moderate to severe exacerbations for the group taking Roflumilast. But if we look at Figure 3, we can isolate the most critical secondary outcome: strictly severe exacerbations. For events severe enough to require actual hospital admission, Roflumilast drove a nearly 24% reduction. This drop carries a dual impact. It improves survivability for the individual patient, while simultaneously lifting a massive economic burden off the wider healthcare infrastructure. Systemic Roflumilast successfully shattered the treatment ceiling, preventing catastrophic hospitalizations where maximum inhaler therapy had failed. Beyond tracking exacerbation events, researchers evaluated physiological lung function, specifically measuring post-bronchodilator forced expiratory volume in 1 second, or FEV1. This line graph from Figure 4 tracks that FEV1 trajectory over the full 52-week treatment period. There is a sustained 56 milliliter improvement in forced expiratory volume for the treatment group compared to the placebo. Mathematically, 56 milliliters is a modest increase, roughly a feeling of breathlessness, but its presence is a vital biological marker. This FEV1 bump proves that Roflumilast possesses an orthogonal anti-inflammatory action, functioning independently of the background bronchodilators the patients were already taking. However, the reality of pharmacology is that measurable systemic potency often carries a corresponding biological tax. Table 3 catalogs the adverse events profile, with higher patient withdrawals in the Roflumilast group, driven by gastrointestinal disruptions like diarrhea. Table 4 reveals a specific metabolic toll. The treatment group experienced an average weight decrease of 2.65 kilograms. Unintended weight loss, known as cachexia, is a particularly dangerous complicating factor for patients suffering from severe COPD, who already struggle to maintain muscle mass. There was one crucial negative finding regarding safety. Unlike high-dose inhaled steroids, systemic Roflumilast did not increase the incidence of pneumonia. While systemic Roflumilast is a potent tool for failing lungs, its gastrointestinal and metabolic side effects limit its broader application. Which brings us back to the opening paradox. Why take a heavy-hitting, GI-disrupting COPD drug and use it to treat flaky, inflamed skin? By utilizing a targeted, localized topical delivery mechanism, the drug successfully neutralizes dermal inflammatory pathways while completely bypassing the bloodstream entirely. Formulating Roflumilast as a 0.3% topical foam captures the exact PDE4 anti-inflammatory power proven in the REACT trial. Because the foam formulation avoids gut and liver metabolism, it effectively erases the systemic risks of diarrhea and cachexia. The route of administration serves as the ultimate pharmacological filter, stripping away systemic toxicity while preserving the core mechanism of action. We can trace the evolution of Roflumilast from a last-line systemic defense for severe pulmonary disease, transforming it into a frontline topical dermatological tool. The 2015 REACT trial demonstrated the molecule's ability to maintain its effect under the heaviest possible biological stress testing. Identifying the correct inflammatory pathway is a partial solution. The delivery route determines whether that potency remains clinically tolerable. Pharmacology achieves its clinical goals when we understand exactly how a drug works and control precisely where it travels.